Happy Thursday morning, and this is the first of our two live online sessions that we will have this week, covering the material for the week. I believe it's a week 11 now, and on this week's material, we'll be looking at water, fluid balance, electrolytes, pH balance, and things like that. So it's a little bit of a continuation of what we saw last week when we looked at the urinary system. And now we're especially focusing on the physiological side of the house, but we're looking at the physiology of maintaining a water balance in a larger scale, considering it on the scale of the entire body. Before we get into the material that I have, I wanted to share quickly a plan for today's session. And as previously, I will be offering timestamps in the description for this session in case you're looking for a particular part of the information. So a few things that we'll do today. We'll start, first of all, by covering what it is that I'm expecting you to do this week. There's going to be few differences in terms of the workload and assignments from what you've seen before. Secondly, I want to go through the lecture material, but we're going to keep this as a very general, just an overarching quick review of the chapter material as it relates to this week's content. And finally, as a third element, I want to spend a little bit of time talking with you through the lab which we have for this week. And this week's lab is going to be somewhat familiar to what you've seen in some of the previous labs where we do kind of case scenarios, but it's going to be a little bit of different in the scope of it. So those are going to be the three things that we'll be looking today. And to make a start with the first element for this week's live online session, let me share a screen with you. So at this point, you should be seeing my screen from the canvas, and this screen really shows you a student view of what you should be seeing roughly when you go to the learning modules. And we're just going to keep scrolling down until we get to this week's material, which is going to be the week 11, looking at the water, electrolyte, and pH balance. And as always, I have provided you the lecture slides. I provided you what I consider is a nice, a little more in-depth review of the chapter material as a link to a YouTube video that I prepared for you. And we also go through the assignments for this week. And the assignments that I have for this week are actually only two of them. So because we're talking about a physiological set of systems, there's not going to be practice anatomy that I would ask you to complete. So there's simply going to be the quiz, which is a similar format of a quiz that you've seen before. And then there's going to be our lab, which is going to be a critical thinking assignment. So only two elements that I'm asking you to complete this week. So please make sure that you get those done, and we'll get you on a path of getting those points and being successful on the course in that sense. Another thing that I wanted to give as a quick shout out is that this is going to be the last chapter that we cover before next week. We will be moving on to the exam 3 and lab practical 3. And we're going to do the same format as we've done before. I will be providing you with a study guide. We will go through that in a classroom setting, and we'll set you up for success for both the exam and lab practical. The reason why I wanted to bring it up at this point is that keep in mind, by the time you're finished with next week, you have completed 3/4 of the material for this course, and you will have approximately 3/4 of the final grade for your course grade as well figured out. So that's pretty exciting. At least I think that it's highly motivational to know exactly where you are standing with the workload at this time. But really, the big take home message that I wanted to share at this first part of our time together is that just simply remember you only have to complete the quiz for this week in addition to the lab. And I receive every now and then requests to have some of the assignments opened, and you know that I'm trying to be very, very flexible with those. So if you have sent me a message and asked any assignments to be opened, even to be given an extension for any of those all I should have been able to process at this point, and all of them should be open. If you need additional time, do not hesitate to message to me. I want you to have every chance to collect those points towards your final grade. And now I'm going to stop sharing that screen, and instead we will jump on to the second portion of this week's content that I wanted to cover with you. And that's going to be the lecture kind of a portion, just going really quickly through the key points as it relates to the water, electrolyte, and pH balance in the body. Please remember that I do offer a full length lecture video on these topics, and that's available for you through your Canvas course. So today is simply going to be a kind of a quick summary review of all that. And like I said, this is rather continuation, I would describe it, for what we saw last week when we talked about the urinary system. But this time around, we're taking the focus away from just one system and looking at it more holistically throughout the entire body. How do we manage the water balance? How do we manage the electrolytes? And how do we manage the pH balance in the body at the large scale? And we're going to see, especially when it comes to the lab, that there's going to be so many systems working together to accomplish these goals. So it's kind of a nice way to move away from this systems approach and have a bit of a more of a whole body approach on the topic. When I was preparing for this session, I was thinking, how do I convince you that this chapter matters? And I think we often go back to what is your ultimate goal? And many of you are thinking a career in allied healthcare fields, either in nursing or other related fields. And of course, one of the things that we end up seeing is that you will be getting on those fields various lab reports back to you. And these lab reports provide some important information. They can be about the blood or urine, but they provide information such as how is the state of the fluids in our body, how about the electrolytes, other dissolved components in these fluids, and of course, what's going on with the pH balance as well. And if any of these is not where it should be, that can result in the patient being very unwell, or in the worst case scenario, it could lead into the death. So definitely, I think, an important topic for us to discuss. If you've taken BIO181 with me or someone else, you probably will have heard this short spiel that I'll share with you. I use a picture of the Earth on my 181 class to just make an opening about the fact that often when we talk about the Earth, our planet where we're living, we talk about the blue planet. The name gives us a clue that what's going to be important for us is going to be all the water. And even on this picture, we see really nicely the huge amount of the surface of the Earth that's covered by oceans and other large bodies of the water. We see some of the water also in the atmosphere as clouds and so on. So water really is highly prominent on Earth. And that gives us a clue that water is really the essential thing for the life. If we use our best knowledge, and of course knowledge changes always, but based on our best understanding at the moment, without very few minor exceptions, all living things require water for life. All living things are largely made of water, and based on our best understanding, life also evolved in the water. So this is the reason why when we go to this space and we start looking for potential whether a life exists or has existed before at another planet, we definitely end up looking for evidence. Is there water or has there been water on that planet? Because without water, our chances of finding life are lesser. So that really just, I think, hits home the importance of why we're going to spend a little bit of time talking about the water. Another thing, just as a side tangent to this story, to keep in mind is that I find it amazing that we're doing such a great job of going to this space in the areas that are close to our home and then to the deep space missions even. But yet we don't fully know our own planet yet. What's deeper at the deepest parts of our oceans is still largely unmapped. So we're doing a great job of investigating a lot of things even when we don't know our own planet yet. So just putting it out there as a thing that's very, very interesting. So like I mentioned, most living things are made of largely of water. So this can be anywhere. In case of humans, you see different values in different textbooks. Somewhere in the range of 60 to 70% of you will be water. And actually the percentage number varies a little bit depending on a few different factors. There can be things like body size, sex, a level of activity, age, lifestyle, environment where you live. But on average, we're looking at numbers somewhere around 60 to 70%. What's interesting to me is that there is going to be a sex difference, with the males being a little bit more in terms of their male content being a little bit more made of water than females. And this can be explained by the higher body fat percentage in the females and males having a little bit higher skeletal muscle mass, which both contribute to this total water amount in the body. Another interesting thing that I find kind of fascinating is that infants, newborns, babies, they have a little more water in their body than when you look at the old age, the water content drops. This is really to be explained, and especially in the case of the infants, the low body fat percent and low bone mass and things like that. So there's a lot more to the story. I think if you're thinking of the exam that's coming up next week, I do like asking that if I have these 4 groups listed, that on average, typically, which ones of these would have most water and least water. So we're going to see infants having most water, then males, adult females, and then elderly people. I don't know if that was totally fair. I was trying to be funny, but maybe it wasn't the nicest slide that I could come up with. Grapes turning into raisins. But whatever helps you to remember the concept, right? So I guess that the next question would be that where in your body is this water located at? And we end up noticing, please keep in mind that these percentages are referring to the total adding up to 60%. So they're not absolute numbers. Where in your body the water is located, about 2/3 of all the water in your body is going to be what we call intracellular fluid. And intracellular fluid refers to the fluid, the water inside the cells. So most of the water that we find in the body is going to be inside the cells. a portion of the water, about 1/3 of the remaining water, is actually located outside the cells. So that's known as our extracellular fluid. And you might wonder that where in the body would this extracellular fluid be? Well, there's going to be 3 groups. So first, I wanted to share the interstitial fluid, which is going to be the fluid located between the cells that we're seeing. Outside the cells, between the cells, filling those gaps in the tissues. The second and third group of extracellular fluid that we're going to see are going to be familiar to you from our previous discussions on this course. So we're going to have, of course, water as a content of the blood. To be more precise, the plasma of the blood. And we're going to see also water within the lymph. So blood, plasma, and lymph form the second and third group of the extracellular fluid and where we might find that. So whenever I talk about topics and we go on to and it's some of these basic themes, I always end up thinking that, am I doing a good job showing you why this matters? So what I'm going to share next is not going to be in the exam. It's simply to show that this extracellular fluid is going to be of an interest to us. So I'm going to share a little bit of my work in the research field and how it relates to this. So like I've shared with you in the past, I've done a lot of work with MRI. MRI is particularly good for imaging technique for soft tissues. So here we're seeing an image of a heart. We see the left ventricle. Notice the thick muscle wall there, and we see the right ventricle thinner wall there. We see the chest, some adipose tissue on the surface. We see liver underneath the diaphragm and the lungs. Keep in mind that this is going to be a little bit from an odd angle as a section, because what we're doing here, we're looking at the heart in relation to the heart's own axis. And remember, it was located in this slightly awkward angle in the body, so it doesn't align with the planes that we've covered before. It's going to be a little bit of a different oblique plane. So this is a typical image that we can acquire with MRI. And what we're seeing here, we're actually seeing some nice papillary muscles, so structures that we have covered in the past. These images are acquired by using a contrast agent, doing a few rounds of pictures, then superimposing those and so on. And what we're seeing is that on gadolinium enhanced imaging, which is the kind of a standard gold practice that we've used for the past, what would I say, 10, 15, 20 years for taking images of the heart and looking at the heart wall injury, that the dead necrotic tissue in the cardiac muscle wall will show up as a bright signal. So what you see here as this white dot, that's actually still going to be part of this muscle wall that we could see here, but this is now dead tissue. It has gotten damaged and it's not going to come back alive, even if we do revascularization of the heart. That area has been lost. So of course the question then comes is that, could we do better? And when would it be useful to do better than what we traditionally do? And the group that I've been part of working on, we're curious of, can we predict, rather than showing what's dead, what will be dead? What area of the heart wall is at risk of being damaged if we do not do something to treat this patient, such as a revascularization of that part of the coronary wall? And turns out with some clever computing, some clever work with the MRI scanner, that is possible. And what you see here are known as our T1 and T2 images. They're a little bit of a more advanced process, superimposing data again, doing some more advanced computing and so on. But all of these images are from the same patient who, as you recall here on the picture A, had dead muscle wall here on this picture. But you'll notice, and please don't worry too much about the colors. Colors are there just to communicate things. What we're really doing is we're looking of signal intensities as a number value, not so much what color we're seeing when we're analyzing these images. But you'll notice that on the image B, the area that shows up bright is going to be significantly larger than what we saw here with the image A. And same goes for the image on the labeled as image C. So the bright area on images B and C actually symbolizes the area where we are expecting that it's an area at risk if we do not do anything to salvage this. What area will die, become necrotic because there's not sufficient vascular supply to this area. Well, you might wonder, how do we know that this is going to be the area that's going to be damaged? Because knowing this area would be highly useful. If you think about it, any procedure has risks. And we want to balance that concept of risks for treating someone versus the benefit of the treatment being successful. So we're wanting to weigh in, is it worth of doing this treatment for this patient, considering that the treatment carries risks? So the way how we can acquire these area at risk, the potentially the area of the wall that potentially might get damaged, is actually by looking at the amount of water concentration outside the cells within this tissue of the cardiac muscle wall. So this is going to be the extracellular fluid. And what we have found out from the lab research is that the volume of the extracellular fluid increases at the area that will eventually die before the damage is irreversible. And we can pick up this increased water content greatly with MRI scanners. So this is one clinical application of what you're learning about the very basic things. So simply by knowing from the lab research that ECF increases in the tissues before the tissue damage is irreversible, if there is myocardial infarction, that helps us to apply this basic concept to something that can really make a huge difference in a clinical decision making of whether an invasive procedure is useful or not. So as I said, I would never ask you to be able to repeat or recite this story in an exam. I just simply wanted to show you that ECF measure does matter in a clinical practice. So having said all that, I think it's time that we move on and we discuss about the water a little bit more and about the characteristics that make water so important. And again, I believe that you would have covered these characteristics of water in your Bio 181 or 156 class that you might have taken as a prereq for this class. But I think that it's still worthwhile that we at least mention. So the first thing on my list here is going to be that water is great at transporting different materials. So let's think of, for example, humans, and it's going to be the blood that's going to be our transport media. And of course, blood was largely made of water. And within the blood, we're able to transport things such as respiratory gases, nutrients, waste products, hormones, certain components that we remove with the urinary system, and so on and so on. So a lot of things can be transported with the water component of the blood. But it doesn't just have to be the blood. We're going to have, if we look at comparative anatomy and physiology, certain insects that don't have blood, they have a different kind of a fluid, but the fluid does the same thing of transporting materials. Or in this case, that I've included a picture of a plant, it's not just the water that we're trying to extract from the soil, it's the nutrients that get carried with the water to the tree and transported to various different parts of the plant where these nutrients are then used to do various things that get done, for example, doing photosynthesis. So water really is highly successful in transporting materials. And that's one of the key reasons why water is the foundation of that living creatures, living things rely on for transporting the materials on these organisms. The set of next two terms, cohesion and adhesion, I believe you are already familiar with, but I'm just simply going to, as a reminder, share those. So the term cohesion referred to the attraction that water molecules had with other water molecules. And you remember, of course, and again, this is not something that we would go into this detail in our exam, but you remember that this was because the water molecule shared unevenly the electrons. So there was a partial charge difference, and these partial charge differences from one water molecule to another caused this bonding pattern, hydrogen bonds that we saw and we discussed. So cohesion, remember that, referring to the fact that water molecules are attracted to each other. The term adhesion instead refers to the fact that water molecules tend to stick to surfaces, and this is especially to polar surfaces. And by polar surfaces, I mean that surfaces that carry either positive or negative charge, water molecules tend to stick to those as well. And some of the examples of that we can see here, for example, we see nature, cobwebs, things like that, water droplets attaching there and forming larger droplets as one water molecule is attracted to another. We can also see this if we look at a drinking glass. So this is just a side view of a drinking glass. And when you have some fluid in there, let's say that it is now water, and some of it is used, we can see this concept of adhesion sticking to other surfaces in the water, kind of climbing a little bit up on the edges, on the sides of the glass. As you keep drinking more and more out from that glass. So that's the adhesion sticking to the walls of this glass that we're using as an example. Another example, and actually an example of cohesion, would be how water can sometimes, let's sketch another diagram here. If we have again a side view of just a drinking glass, this time it's bang on from this side, so I'm not going to draw that circular top. And you can actually overfill the glass of water so that it starts to dome up a little bit. So kind of come a little bit higher where the highest rim of the drinking glass is. That's because these water molecules that form that dome are still attracted to each other, so they don't give up and just flow out. So you can kind of overfill the glass a little bit as well. So that would be an example of adhesion. Keep that in mind when next time you're filling in your drink cup at the gas station. Just a joke. Kind of tying into this concept of cohesion, is going to be our next example, which is going to be that water has high surface tension. So water molecules were attracted to each other, and certain animals, such as insects, are able to use this, because this results in what we know as surface tension, the surface of the water being able to carry their weight, and these insects rely on traveling on that. Actually, you might have seen examples of a high surface tension of water if you've done jumping into the water from a height, and especially if you've done it as a belly flop, belly first. Water surface can feel really tough and hard to fall against unless you break the surface tension first, for example, by going feet in first or hands in first. So you kind of ease some of that surface tension. If you just fall flat on the surface of the water, water can actually be really, really hard surface to fall on. So that's another thing to bear in mind. Another thing, and it's not going to be a real estate class, but I will use real estate as a little bit of an example. So people typically, and animals as well, are preferring to live by big bodies of water. The coastal areas, whether it is an ocean, sea, lake, or whatever, we have this tendency of preferring those areas where we have a lot of water around as areas where we like to habitate. Well, why is this the case? Well, let's think of that a little bit. So I'm drawing a graph. This is going to be time on the x-axis, and y-axis, it's going to be temperature. And if we look at a temperature just somewhere where it's very dry, let's say Arizona, but we'll notice that during the day, the temperatures on a summer especially climb up, and then during the night, the temperatures drop down. So we're getting a lot of fluctuation on the temperatures throughout the day, just simply because it is so dry. However, now, if we move on to live close to a big body of water, one of the things that we'll end up noticing is that water during the day is able to bind some of that heat, and then during the night, it's able to release some of that heat. So it evens out some of these extremes because of water's great ability to bind and release heat as needed. So that's why on the coastal areas, the temperature fluctuations are not as drastic, and we tend to prefer that. One thing that I was never prepared when I moved to Arizona, I knew it would be hot days. I didn't quite realize how cold nights it might get to. And that's simply because we have very little humidity. We have very little moisture around us. Whereas somewhere on the coastal area, you get these more consistent temperatures with lesser extremes. So this concept that water is great for binding heat is not just used in real estate. We see this in biology. For example, we can use blood, which is largely made of water, to transport heat from one part of the body to another part of the body as needed. So this can be, for example, if we're out in a cold environment and we would be worried of getting a frostbite on the ears, nose tip, fingertips, things like that, we simply carry blood that's nice and warm from our core to these parts to bring some of the heat to those parts. Another example of water, and in this case, water in blood being used to transport heat, would be when we're doing a lot of exercising, for example, and we produce heat as a byproduct of that muscle activity, and we want to get rid of that excess heat. We carry that heat with the blood to the surfaces of the skin, and then we can just sweat it off, and that heat we can get rid of that through processes like evaporation and so on. So that's the fifth element on this list of characteristics of water that make it so ideal for life and maintenance of life. What I have next is just a reminder, mainly because I use these slides in BIO201, is that, of course, water can take three different phases. It can be gaseous, kind of water vapor. It can be liquid, just the water in a liquid shape that we often, for example, drink, a liquid shape that it travels in our cardiovascular system and so on. And water, of course, can take also solid form when it freezes. We discussed about gyrogenic freezing of the bodies in a classroom and how that would have some of the problems, because as water freezes, it also tends to expand, and this could be problematic for the cells and damaging cells, for example. And for this reason, why we need to then think of, okay, how do we prevent this damage from happening? There are animals that are adapted to this and produce chemical substances that help to deal with that. But that wasn't really the main focus of our chat, so I'll leave it for the classroom discussions. I also mentioned plasma as the fourth phase of materials that they can take. Of course, that's not going to be the case in the case of water, but just for the sake of completedness, I included it there. I guess the most important characteristic of water that's going to be really, really important for us, why water is so good for our ability of having living organisms rely on water, is that water dissolves many things. And in fact, water is known as a universal solvent. So we see that because a lot of things can dissolve in water, water can then be used to transport these things from one part of the body to another part of the body as needed. So those two go kind of almost like hand in hand. There are, of course, going to be certain elements that do not dissolve in water, such as glucose, lipids, urea, creatine, and so on. These are not as much of an interest for us, even though they don't fully dissolve, they can be still transported in this heavily water liquid from one part of the body to another. But what's going to be more important for us for the purposes of our chat today are going to be these materials that dissolve in water, such as the various different kind of salts, acids, bases, proteins. And as these things dissolve in water, you'll notice that they form various different kind of charges. And these charges were essential for us when we considered that this is what we used to drive many processes, many things. So some of these we do find lots in various different body fluids, for example, in the blood or in between the cells or inside the cells. Let's see a couple of examples. So sodium and potassium that I have here. Remember the sodium-potassium pumps that we had on the surface of the nerve cells, neurons, to establish this imbalance between outside and inside of the nerve cell, inside of the nerve cell being slightly negatively charged, about negative 70 millivolts. So it's simply by moving these charged particles and putting the energy on pumping them that is essential for how nervous system works and neurons work. Other examples, what do we have here? Let's have a look of another good example, our chloride, bicarbonate. We've just spoken about bicarbonate when we talked about the transportation of carbon dioxide in the blood. Various different kinds of hydrogen derivatives as well that we see there. But really, if nothing from this last part of the discussion, what I'm asking you to take away is that wherever water goes, the electrolytes tend to follow. And the other way around, wherever electrolytes go, water tends to follow. So water and electrolytes are connected to each other when we look at the physiology. So we could say that they are interdependent. So there is a connection between the two. Where one goes, the other one will go. And that's actually going to be one of the exam questions. And we'll end up seeing some examples of this interdependent characteristics of water and electrolytes, so that one is dependent on another. When we talk about effects that we might have clinically on someone who sweats a lot and tries to replace that lost fluid with water but lacks electrolytes, how that can become a problem as well. So we'll see some examples of that in just a bit. I guess the next thing that I thought that would be nice to speak about is that how much water should you consume in a day and how much water do you lose in a day? Well, in an ideal situation, these two are going to be in balance. So how much water you consume is an equal to how much water you're losing. And if it's in a balance, we're in a good place. Of course, otherwise, you might end up gaining too much water or losing too much water, getting overly hydrated or dehydrated. I've marked there, and of course, this is again dependent on an individual. We're seeing a lot of individual variation based on body size, sex, activity level, environment where you live, things like that, body composition even. But on average, roughly a human being should drink about 2.5 liters of water in a day. How much that is in a gallons, I'm not great at metrics, at other systems than metric systems, so I guess that it's a little less than a gallon, but it's a lot of water. I don't think I'm very successful of being able to consume quite that much water. So many of us could do with a little bit more water consumption than what we're doing at the moment. Well, what is the force that drives you to get more water in? Well, it is going to be the thirst. So through the process of drinking, we get a little over half of the water that we get into our body, and then the remaining roughly 1/3 through food items, food material that's moist as well. You might notice rather curiously that I marked there that metabolism results in about 10% of the water intake in your body. Remember that chemical formula that I've been writing now time and time again on the board when we've been talking about cellular respiration, how we take sugars and we take oxygen and we convert that into ATP and as a byproduct to carbon dioxide and water. This is the water now that I refer that is the byproduct of metabolic processes in your body. So that contributes about 10% of the total water intake to your body, but mainly through the drinking and then eating moist food stuff. Well, how do we lose water from our body? It's going to be mainly through the process of urination. Again, more than half of it, just the urination. There's going to be about 1/3 that covers a larger group of things like evaporation, sweating, the humidity we lose through breathing. So either through skin or through the respiration, we also lose a good amount of water. Very little water is lost through the feces, and we'll see this in our next topic of interest in a couple of weeks when we talk about digestive system, how your large intestines are excellent at recovering so much of the water from chyme before it becomes feces. So we're very, very, very successful in that. So how do we regulate the water balance in your body so that the water intake and water output would be even, that they would be in balance? And you're going to see many different kinds of diagrams depending on what textbook you use. Some of them are more complex than others, but multi-step processes. Some of them are a little bit more like a seesaw. So there's two opposites, overhydration and underhydration, or they might be loop-like diagrams. But if you remember and go back to the beginning of our course, I said that as long as you learn this concept of homeostasis and homeostatic feedback mechanism loop, you will be fine in my classes. So we come back to this again. So homeostasis referred, of course, to the maintenance of the consistent internal environment. And here we have the loop mechanism. So something changes, we detect that, pass that information to processing and compare it against what would be the normal range of values. And if it's off from the normal range of values, then we act on it, typically through negative feedback mechanism that stops the stimulus from happening. So let's have a look of maintaining water balance in body by using this simplified diagram. So we don't have to go through all these complex box by box by box diagrams instead. So in this first scenario, we're going to have too much water around. And I know this is not very scientific expression, but we can think of it as if the blood being too watery when compared to what the normal values are. So this is compared in the groups of cell bodies at the level of hypothalamus, known as osmoreceptors. We're having a look that, okay, we're detecting that now there's too much water in the body. We're comparing it to what it should be at the level of the brain, and we act on it. How we act on if you have excess water in your body is through pituitary, releasing the hormone ADH in smaller quantities. Pay attention that this is going to be now less ADH being released. And you remember that what ADH stood for was anti-diuretic hormone. So it's the hormone that makes us make less pee. So now we're making less of this hormone that makes us make less pee. So now more water leaves the nephrons as urine. So we end up producing more pee, and we simply urinate a lot of that excess water out from the body. So that would be an example how we deal with excess water in your body through the homeostatic feedback mechanism loop. Let's have a look at an opposite event of this. And in this opposite event, now we're going to have too little water. So we are dehydrated. Again, very unscientifically, we could say that your blood is too salty. So again, that's detected by osmoreceptors at the level of hypothalamus compared against the normal values in the brain. And now we notice that we are dehydrated. So how might we deal with dehydration? Well, now pituitary produces more this hormone known as anti-diuretic hormone. You remember what anti-diuretic hormone did, and we saw this in our chapter about the urinary system, and when we looked at the nephron function is that ADH turned the distal convoluted tubule of a nephron more permeable, so that more fluid could be reabsorbed back to the body from the nephron. So we're taking away some of that fluid that would otherwise become urine from the distal convoluted tubule by turning that wall very permeable. So as a result, we're now making less of that output product, which was, of course, our urine. So we're going to make less urine, less pee, in response to this lack of water, in response to this dehydration that we had going on. So I hope that if nothing else, that helped you to realize how these complex diagrams can be summarized in a very good way by going back to the basic concepts. This is why we took all that time to learn the homeostatic feedback mechanism loop, because it really is a useful way of explaining these concepts. I wanted to also talk a little bit about the tonicity, and I assume that this is a fairly familiar concept to you from your BIO201, at least in the class. I was getting a lot of nods that this is something that you know about. So there's going to be 3 cases that we're going to look at. And we're going to start with hypotonic conditions. But before we do, remember what we're seeing here is that materials move from areas of high concentration to areas of low concentration through a passive process known as diffusion. So in this case, we're looking where does the water go, because the water will follow where the electrolytes are, trying to make sure that both sides of the cell membrane have same concentration. And as we'll see, sometimes this task is doomed from the get-go, but we still try to do it. So in hypotonic conditions, the inside of the cell, which I have colored blue in here in this diagram, contains more electrolytes, or you can think of it as being more salty. So water tries to even out the concentration of salts on both sides of the cell membrane, so inside and outside of the cell. And how we try to balance this concentration difference is that we direct water into the cell. We're hoping that we're able to dilute the contents of the cell to be equal to what it is outside the cell. So water is now flowing inside the cell. And water will keep doing this, flowing inside the cell. And as it keeps doing that, it causes the cell to swell, grow bigger, and eventually, in an extreme case, the cell could even burst, break, because of this excess water just keeping going in and in and into the cell. Not a great way to go, I'd say. That's not my plan, at least. So how do you remember hypotonic cells? As always in A&P, relate the terms to something that is easy for you to remember. When I hear hypotonic cells, I think of this animal known as hippo. Hippos are these kind of a big, swollen-looking animals. So hypotonic cells are hippo cells in my mind. They're these big, swollen cells because water is now flowing inside these cells. So that was our first case. Let's have a look at the second case. In the second case, we're looking at isotonic cells. Isotonic cells are a situation where we have same concentration of salts or electrolytes both inside and outside the cell. Because the concentration is the same, even though some water will come into the cell, some water will also leave the cell, and there's no net gain or net loss of water for the cell. So the cell remains the exact same size. There's no change in the cell size. So how do I remember isotonic cells? Well, I think of these cells telling me that iso perfect. I don't need to change. I'm exactly what I need to be. So my isotonic cells are iso-perfect cells. They're perfect as they are. They don't change in the size at all. There's no net gain or net loss of the fluid in those cells. The final condition that we're going to see is going to be our hypertonic cells. In a hypertonic situation, there's going to be more electrolytes or more salts outside the cell. And again, water tends to move to the direction trying to even out these concentrations. So water is leaving the cell, trying to balance off this excess salt concentration outside the cell. And as a result, the cell keeps drying up or shrinking and might eventually even end up drying up. So how do I remember cells that are hypertonic cells? Cells where the water is flowing outside from the cell. Well, I think of what kind of people are people who I describe as being hyper. These are people who have a lot of energy. What do you do if you have a lot of energy? You run around a lot. Well, what happens to you if you run around a lot? You become very skinny. So my hypertonic cells in my mind are skinny cells. They are shrinking as a result of water leaving the cell. So I hope that that was a review that helped you to remember those three conditions. And at this point in the classroom, we kind of practice that and consider the case where you can actually die as a result of having to consume too much water in a short period of time and not removing the excess water through the process of urination. There was an unfortunate case of that where a radio station organized a competition where they challenged participants to drink a lot of water in a short period of time, not use bathroom in an bid to win the Wii game console. I think the competition was called Hold Your Wii for a Wii, and that didn't end up well. We had a loss of life because I guess the moral of the story is that everyone should do some basic A&P classes, even disc jockeys on a radio station. No, but to be serious, very sad case, very unfortunate. So a good example of why we need to listen our body. If you need to urinate, you probably should go and do that. Kind of tying into what we've been just looking at, I have one more example here of a situation that I at least can relate to, that I'm familiar with. When you're out on a hot Arizona summer day, you end up sweating a lot, and you might try to compensate for this loss of fluids by drinking a lot of water. Well, what happens when you drink a lot of water and it's just water? Your body fluids become too dilute, and your brain, through those osmoreceptors at hypothalamus, will now send you a command to urinate, and you will actually become more dehydrated simply because you were drinking a lot of water. So it's almost like self-fulfilling prophecy. And what you're trying to do works against you. Because in this case, if we're losing a lot of electrolytes through sweating, just replenishing the fluid concentration is not going to be enough. You should drink something with electrolytes. So often in these cases, we get a craving for salty foods and stuff. That's your body trying to get those electrolytes into your body. So individuals who do high performance sports, extreme athletes, they actually don't consume just pure water at their peak performance, but they use special recovery drinks or exercise drinks that contain high amounts of electrolytes, so they can replace those electrolytes that they lost through the sweat during the exercise. And that will help to enhance the water uptake to the body and to the cells. Some of the drinks, I compared them a little bit here. I did a little bit of research on that. I guess the moral of the story is that soft drink sodas are not great, neither are juices for replenishing these electrolytes that you lose. So that's why we have these electrolyte drinks. They should not be day-to-day drink. They are for a specific purpose of replacing lost electrolytes in individuals for who this might be an issue. So having said that, I think the third and final part of our lecture part that we were supposed to look at is going to be all to do with body pH. So let's have a look at that a little bit. You remember pH scale running from 0 to 14, around 7 being the neutral. You could have higher values of pH telling that something's acidic, or lower values of pH telling that they're basic substances. We're looking at the concentration of hydrogen ions. So this is really where just even the symbol of what we know as pH comes from. We're looking at the hydrogen concentration. And if my memory serves me correctly, this is not going to be actually a linear concentration. It's going to be more of a exponential curve. So it's not that you can say that we're going to have twice as many hydrogen ions on something that has a pH of 4 in comparison to pH of 2. It's a little more complex than that. But the take-home message here is that we're looking at the concentration of hydrogen ions. Well, why is this important for us? Why do we care? Well, we care because actually your body does a remarkable job of maintaining a fairly consistent normal range for your pH in your body. Normal pH in your body is somewhere around 7.35 to 7.45. So only 0.1 of a unit's variation. That is a very, very small variation. And you'll actually end up seeing that if you ever work in a lab and try to mix different substances so you could get an end product that has a specific pH. It is hard to do, and yet our body does it so remarkably well. If you look at the survival range even, if your pH is below 6.8 or above 8, you probably will be very, very sick, or you might end up dying. So that's still a small range. So there's something remarkable that our body is able to do to control our range and keep it within this small range of values. Well, just before we look how your body does this, I just wanted to share the two conditions, alkalosis and acidosis. If your body pH is off from that normal range, and they can have many different kinds of consequences. So I mentioned that your body does an amazing job of keeping the body pH within this narrow range. And that's actually because of 3 different mechanisms that we have interplaying here. First mechanism that we have is using various different buffers. Buffers are simply chemicals that bind into acids or bases and bring their pH back close to the neutral, the 7. So these chemicals act very, very fast. Within seconds. It's a very fast first line of defense against the pH change. Another way how we can deal with a pH change and kind of respond to that is by using our respiratory responses. So the center at the brainstem for breathing regulation detects the hydrogen concentration, and as needed will then regulate the rate and depth of breathing to deal with that. This is a slower response, takes minutes to act, but has more long-lasting effects than the buffers. And the final mechanism is going to be a renal mechanism, so through the kidneys, and we just had a look of that in the last chapter, how we use kidneys to remove excess hydrogen ions. So I'm not going to go through it again. You can refer back to that video, and I'll link it also to the end of this video in case you missed it. But what we're seeing is that this is a slow process. It might take anywhere from hours, even up to days to effect, but the effects are long lasting. And this is really the most potent, most strongest response out of these three that we see. So these are the three methods, three mechanisms, how we respond to the need of maintaining the body pH within this narrow range that we saw earlier on. And kind of to wrap this up, I wanted to show a few examples of the clinical conditions, acidosis and alkalosis, even though the signs and symptoms start as fairly not dramatic headache, feeling confused, lightheaded, sleepy, confused. The condition can quickly worsen to the level of loss of consciousness or going into coma. There's a lot of things that can go wrong when the body pH is not where it should be. So I really just wanted to kind of highlight it and show it that this is a serious issue. And that's why we have these amazing response mechanisms, the buffers, respiration, and renal mechanism to maintain our body pH where it needs to be. So I want to wrap up this section by talking a little bit about fluid therapy and how that is often the first thing we do when you come to see someone in a medical field. If you're not well, we administer you fluids that contain just the right balance of electrolytes and salts so that it's what it should be. We're not getting any of these issues with tonicity. It's exactly what your body is looking for. So I sometimes call it a little bit arrogantly as expensive salt water. That's not quite the case. This is sterile content and it's ideally balanced. So it is expensive for a reason. But that's one of the first things that we do because we do want to make sure that you're not going to be dehydrated. We want you to have fluids in your body. This helps to bring up also the blood volume, blood pressure, and so on. The other reason is also by having these fluids running into IV fluids, we're able to quickly administer through that many medications and get it to the system rather than giving you a pill and waiting for up to hours for it to act. As a side note, a side tangent, I'll also share that really dehydration is one of the main killers in the world, especially in the developing world. We'll see examples of that when people have diarrhea, how they lose a lot of fluids, and that can be it. So we're very, very fortunate here in the Western world with the medical care that we do have available for us. Kind of one final point that I got some questions from students in the class was that, well, how about when some people eat food things that are either acidic or alkaline, drink alkaline water? Are you able to change your body pH significantly through this? Well, even though there might be slight short-term effects, especially in the case of high-intensity athletes, remember we had these three mechanisms to keep your body pH within this narrow range. Your diet will not change this. These mechanisms will bring your body pH where it needs to be. I think that from my perspective, the preference for following these diets where we eat food products or consume liquids of a certain pH is more to do with the fact that those foods are generally just better for you. Let's say if we're doing acidic diet, eating a lot of citrus, eating a lot of fruits, that's good for you. So it has really less to do with the pH that those substances are, but that those substances are generally good for you, and it's a good rule of thumb to think of them through that pH, but it's not that you're really changing your pH value in the body. Well, I hope that that helped to clear off some of the confusions that might sometimes take place, and you feel better having covered that lecture material. As always in my classes, we also in the face-to-face class took a little bit of time to go through the review sheet. I'm not going to share it here on an online setting for the reasons that I've shared before, and copyright issues and so on. But if you didn't get a chance to see it in a classroom, I'll be more than happy to bring you up to speed with that. And I think that the last thing that we have left for this video is to look through our lab, and we'll have a look of that together also as a recording, just in case if you missed the face-to-face class. But when it comes to the labs, I really encouraged you at the beginning of the semester to get to know your lab group partners, because they are going to be the ones who are going to be working with you throughout the entire semester, and they are going to be the ones that can help to bring you up to speed if you miss any information or any classes. So I have prepared a little bit of a PowerPoint. We're going to look through this, even though I know that this review session is going a little longer than what I'd normally like it to be. I still think it's useful. Like I said, I offer this video through timestamps, so you can jump right to this part if this is something that you're looking specifically. So, to give you background why we're doing this lab, higher education institutions such as community colleges are accredited institutions, and our accreditor is actually going to be Higher Learning Commission, HLC. And as part of the accreditation thing, what we have done is that we look through different things of the institution. What are the learning outcomes for different courses? What materials we're using for that? How do we deliver the instruction? Do we have enough seat time with the students? How are we assessing that you meet those learning outcomes? Other things that go to the institution, the support services, even to the level of financial aid and so on. So that all has to do with the fact that we want to make sure that institutions meet the basic standard level that's expected from them. And that's what the accrediting bodies do. And as part of the accreditation process that we do, we take one assignment from every class that's delivered, and that one assignment on every class looks at a certain aspect that we have set out, or an accrediting body has set out for us as an important overarching takeaway thing from the classes where students attend. And in this class, it's going to be your critical thinking skills. So this assignment was written to test how well are you able to extract information from data presented to you. How are you able to find what's important, what's not? How are you able to draw connections between different topics? How are you able to find links and reason things that you haven't necessarily been told, that you just need to memorize, but make new connections with the material that you are familiar, gain deeper level understanding based on that process. And that's really what this assignment tries to do. We change this critical thinking assignment every now and then. So this is one of the first times that we're running this particular assignment through in our classes, but I think it's a really, really good assignment. This will not have an impact on you in terms of that this is what we use for accreditation purposes. You will complete this lab as you would complete any other lab. There's going to be a little bit of impact on my end of the work. I will do actually grading of your assignment twice. I will look at the same question and give it grade that affects your final score, but grade that also feeds into the combined scores from multiple classes, multiple sections across the college, how well our students are doing at critical thinking challenges. So I have shared the grading matrix with you on the canvas, so you can refer there. What am I looking for? What are important things for you to take away from this activity? And how your score is going to be calculated based on that. And really, I always encourage, if you're being told how you're going to be graded, it's worth of using that. Give the grader what they're looking for. And this critical thinking assignment is based on kind of a clinical scenario where we will look at what's going on with two subjects at 4 different time points. So in this scenario, we're going to have two ladies, Carla and Maria, who are going to be taking a hike at Grand Canyon. From rim to rim, I think it's about 24 miles or so. And during this hike, we're seeing what happens to the body of these two ladies. These two ladies are, for our purposes, exact same age, exact same fitness level, have done exact same training, have same experience. So they're equal in all other aspects of but two variables that we will give. And the biggest variable that we will see that's different between these two ladies is that the other lady will have a water container, CamelBak, that they're holding their water as they're going on this hike, and there will be a leak in this water container. So as a result, this other lady is going to have less water available for her for the duration of this hike, and she will end up being a little dehydrated. The other lady instead is highly worried of becoming too dehydrated, so she's drinking a lot and probably more than she should, so she becomes overhydrated. And we're going to look at various clinical measures that we're seeing and how are we seeing these changes based on this only difference being dehydrated or overhydrated between these two ladies. And we'll look at this at 4 different time points: before the hike, during early part of the hike, during late part of the hike, and at the end of the hike. That what are the changes that we're seeing? So you're going to be presented with some data and asked to analyze that, extract values that are important for it, and then you will be given at 4 different parts of this story changes that are happening, and you're trying to analyze how would these changes affect individuals' heart rate, blood pressure, antidiuretic hormone concentration, angiotensin 2, and aldosterone levels. And simply what I'm looking for from you for this exercise is that you tell me whether values are going up or down or remain constant, and you give me some rationale why those changes are happening. I'm not expecting a great level of detail. I'm certainly not expecting you to write as much as I have provided to point you to the right direction in my sample answers. What I'm looking for is more of for you to come up with a decision, what you expect to happen, and be able to justify that decision somehow. So that's what I'm going for, just rationale behind your decision. So let's have a look. Like I said, when we completed this assignment in a classroom together, students tended to divide into 4 groups, each group looking at one part of this exercise, and then coming back together as a whole class and bringing each other up to speed what they discovered. So in the first part, before the hike, for the values that I provided, I asked you to do 2 things. I'm asking you to find if something's abnormal in terms of the values post-hike data, and I'm also asking you to look at what factors are good indicators of the water balance level on an individual's body. And for your answers, I'm completely fine if you provide your answers by just highlighting, underlining, circling things on the data tables that I provided. You can draw arrows going up or down, whether something is elevated or lower than it should be, or whether it's kind of same where it should be. So let's have a look of that. For the values that are a little off from the normal, you can look at the post-hike data, compare it to the normal values. We're seeing things such as on the Carla, the heart rate being elevated, blood pressure being on the lower side, plasma levels, urine osmolarity being elevated. For the Maria, we will see that their post data shows weight that's a little higher than what we would expect it to be, blood pressure on the higher side, plasma and urine osmolarity being on the lower side. So things like that is what I'm looking for. When it comes to what would be good indicators of individuals' water balance, looking at the sweating rate, heart rate, blood pressure, plasma, osmolarity, blood volume, and total body water content, as well as urine concentration, would be some of the things that would be worthwhile to mention. So I think that what I wanted to do next was to look at part 2, where we have now gotten to the early part of the hike. At about 6 miles into the hike, we're noticing that there's changes, and we're looking at those changes when it comes to the hormones, heart rate, blood pressure, water balance. I'm asking you to identify what change are you expecting to see and why. And in case of Carla, we will see that Carla has noticed that her camelback has a leak, so she has ingested relatively small amount of water. Her total water intake is about 250 milliliters, which is not quite as much as we would like it to be, considering how she's sweating, she's having physical exercise and all that. So I'm asking you to look at these 5 different factors and indicate for each whether it has increased, decreased, or remained unchanged when compared to the pre-hike or resting values, and why that would be the case. Again, remember, your answer can be condensed. It doesn't need to be very, very detailed, as I provide a little bit extra information for my suggested answers. For the heart rate, we're expecting that to be increased since Carla is exercising. So that brings up the heart rate. Same goes with the blood pressure. The blood pressure is going to be elevated to maintain the required perfusion of those tissues that are working actively. In terms of the antidiuretic hormone levels, we expect those to be increased because she has ingested less water than she has lost in sweat. So they are becoming, Carla is becoming dehydrated. So we try to counteract that by increased antidiuretic hormone levels. For angiotensin and aldosterone, we expect both likely to be elevated, and I provided the rationale there. One thing to notice with the angiotensin and aldosterone is that these two often go hand in hand for different reasons, but it's a storyline that you want to follow together. If we look at what's going on with Maria, we end up noticing from our assignment description that Maria has been drinking a lot more often. Her total intake is about 1 liter per hour, so she's beginning to feel a need to urinate. So she's not heavily overhydrated yet, but we're starting to work our way towards there. What sort of changes are we seeing on these 5 different measures in case of Maria? Well, the heart rate is going to be increased because she's exercising. Same for the blood pressure to maintain that increased perfusion to those tissues that are active. Her ADH levels are probably unchanged because it's going to be roughly the same amount of water that she's consuming as the amount of water that she loses in sweat. She hasn't yet been urinating, but it's about imbalance, so no changes on ADH. And for the angiotensin 2 and aldosterone 2, we're expecting both to be likely elevated. And again, remember this interaction between the D and E. So full story, I provided it there for you in hopes that you might find it helpful. Part 3 looks at now as are into the hike, about 20 miles in, and we're looking at these same measures. In case of Carla, what we're seeing now is gonna be a little bit of a different situation. Carla is feeling thirsty due to the leak in her water supply, even though she was able to refill her CamelBak at the halfway point. And she has occasionally had few sips of the sports drink that she brought with her. She has ingested about 1 liter of water and sports drinks since the start of the hike. So in total, in 20 miles, 1 liter of fluids, that's not a lot. She is feeling tired and her pace has slowed down quite drastically to 1 mile per hour. And her sweating rate has decreased to 250 mils per hour. So she's sweating less, she's dehydrated. She has urinated once at the beginning of the hike. That was about 500 mils, and her urine was light yellow. She also urinated towards end of the hike. This was only half of what she initially urinated, so about 250 mils, and her urine was dark yellow. So that's giving us a clue of a lot of things that are going on with Carla at this late hike stage. And let's see these measures, what's going on there. So the heart rate is increased because of the exercising. Also, her plasma volume is decreasing. Sweat loss is greater than the fluid intake, resulting in reduced blood volume and decreased cardiac output. As stroke volume decreases, will increase the heart rate through the baroreceptor reflex. Her blood pressure, I would expect it to be either normal or decreased, depending on if the heart rate can be increased enough to compensate for the decreased stroke volume. Her mean arterial pressure begins to fall when the blood volume loss is more than 20%. Her volume is decreasing due to the loss being greater than the ingestion of the fluids, and blood pressure may be slightly decreased. In terms of the ADH, I would expect that to be increased quite significantly because the sweat loss increases the plasma osmolarity, which will increase ADH secretion. Finally, when it comes to angiotensin 2 and aldosterone 2, I would expect both to be increased further due to the decreased blood volume and decreased blood pressure, which will increase the renin secretion and angiotensin 2 production. And in case of aldosterone, that will then the angiotensin 2 include, as well as the decreased blood volume and blood pressure, will cause increased aldosterone release. Let's have a look what's going on with Maria in this same stage of the hike, towards end of the hike. Maria has read that the dehydration is biggest concern when hiking for long periods of time. So she set an ambitious goal of drinking 1 liter every hour. However, she has not been able to quite sustain this during the entire hike, but she's done her best. She has ingested in total of about 6 liters of water since the hike began. At mile 20, she's developing a headache, feeling a little bit nauseous. Her pace has slowed down to 2.5 miles per hour, and her sweating rate is about the same or maybe even greater. She has urinated twice, 500 mils with each time, and her urine was light in color. So we're seeing a lot of clues in this description of what we might expect to see changing here. So clearly Maria has gotten a lot of fluids, probably a little too much. Her heart rate might likely be decreased. Despite the fact that she's exercising, she has also ingested much more fluids than she lost. So this increases the blood volume. Heart rate may be still elevated, but likely lower than early on. Blood pressure, I would expect that to be increased because the blood volume is increased due to the over-ingestion of water, and therefore the blood pressure will likely increase when compared to the early values. In terms of the antidiuretic hormone levels, I would expect these to be decreased because of the over-ingestion of water, which dilutes the plasma and increases the plasma volume. And both of these factors will end up inhibiting the ADH secretion. When it comes to angiotensin 2 and aldosterone, I would expect both to be decreased. Renal perfusion is increased. Baroreceptor input is decreased. These both lead into the lower renin release and angiotensin 2 production, and also the aldosterone 2 production being decreased. So the final part of this lab asks us to look at what's happening at the end of the hike, at the completion of 24 miles for both of these. Let's have a look at a little bit of the background information that we know. Both Carla and Maria ended up stumbling into a ranger who called 911 for them, and medical professionals take the vitals and other important lab values that you saw as the post-hike data. They also determined that Carla is most comfortable if she's lying down, while Maria is more comfortable sitting up. So I'm asking you for two pieces of information for both Carla and Maria. What are the symptoms and vital signs and urine osmolarity that indicate about her body fluid stages? So you're looking at the data. Which parts of the data are worthwhile to notice, and why do we end up seeing in case of Carla that she has lost weight during this 8-hour hike that she's been doing, and this loss of weight, we would not expect that you're gonna burn some fat tissue in an 8-hour hike. So it has to be due to something else than just becoming really fit and lean. There's something else going on there. So let's have a look at that together. And what's going on there is that Carla is dehydrated, so her rate of fluid ingestion is lesser than the rate of fluid loss through sweating, so her body fluid levels have dropped, and this is what we're seeing as the loss of weight. So she has simply lost fluids a lot. Some of the signs and symptoms that you might be paying attention: high heart rate, low blood pressure, telling us that the blood volume is decreased. Urine osmolarity might be another thing to look at. Looking at the urine levels of substances, the characterization that she feels dizzy, tired, weak, nauseous. They're all related to inadequate perfusion of the brain and other tissues as the blood pressure is falling. And also they tell us about loss of water from the cells and cell shrinkage, as we saw when we talked about the tonicity. Because of this, we're seeing issues and hypertonicity taking place. So that was Carla. She was dehydrated. Let's see what's happening with Maria. And same two questions for Maria. Well, we'll end up seeing that Maria has now what we would describe as water intoxication. So she has ingested a lot more water than she has lost or that she's consuming. So that's why she's actually getting weight gain. It's water that she has retained. And some signs and symptoms, we can look at the heart rate being quite low for someone who's exercising. So that's giving us a clue that there's something going on there. Blood pressure is elevated more than we would expect it to be, considering that she's been sweating and losing volume for the last 8 hours. We'll also see in terms of the urine osmolarity that's very low. So she's exerting free water. That indicates that the plasma osmolarity is low and ADH levels are low due to the excess water ingestion causing dilution of the plasma and interstitial fluid. All of these symptoms, headache, vomiting, lethargy, confusion, they do indicate about hypoosmolarity, so hypotonicity of the extracellular fluid, causing a shift of water into the cells and cell swelling. This we saw an example as well when we discussed the tonicity in our earlier part of this exercise. So I think that this is a rather good activity. It pushes you to approach a lab in a little bit more of a different way and really do a lot more reflection. It'll push you to not just look for what you can draw direct connections, but do a lot more thinking yourself. So I hope that you found that interesting and you enjoyed that. And that is where we're going to be concluding our review session today. Depending on whether anyone shows in the evening session, I will repeat this session or parts of that. I will also record it. But if no one shows up, I'm simply going to make this recording available for you because we covered so much on it, and I'll have those timestamps that you can jump into right to the part that you are interested, so you don't need to watch the entire session, so you can find what you want to find most conveniently. As always, I greatly appreciate you being part of this class, and I greatly appreciate all your hard work in this class. Remember, you're about 3/4 in, so the finish line is starting to show up there on the distance, and we'll make sure that you finish off strongly and get that great grade in this class that you deserve. Again, thank you so much for being part of this class. It totally is my joy to work with you. And as always, I am here every step of the way. Do not hesitate to message me. The best way to get hold of me is usually through the Canvas messaging. I have some reference requests, and I'm so happy to help with those. I have had some questions of careers advising and so on. So let's see what we can do and how we can help you the best possible way. I'm here every step of the way. I appreciate you, and I hope you have a very good rest of the week if I don't get to see you again until next week. Next week, we will do the study guide, review the material, prepare for the exam 3 and practical 3, and make sure that you can ace those as well. Until then, thank you and bye.